Cell membranes are very important structures that we're going to find around the outside of every single cell. And if we focus specifically on eukaryotic cells, these are also going to be found in a lot of places inside the cell as well, and they would be surrounding those individual organelles. So as we talk about membrane structure, there will be a number of important molecules that are going to make up that membrane. So if we point them out in this picture here, the first ones, all of these blue molecules, these will be our phospholipids. So we will be reviewing the structure of phospholipids. We're also going to have a number of proteins, and that will be the green structures, both this one here, and then we also have this one down here, which is also representing a protein. They're different kinds of proteins, but still proteins nevertheless. And then the purple molecules that we see here, these are going to be carbohydrates. So let's talk about what each one of these is going to do for the membrane. The phospholipids are going to make up the real structural part of the membrane. So if we review the structure of the phospholipid, here we have an example of one. The main pieces that we're going to have is, remember that we do have a glycerol backbone. And so the glycerol backbone in this image is shown in pink. We are going to have two fatty acid tails. And then also up here, we're going to have a functional group. We're going to have a phosphate group. And then the part shown in light blue here, this is really an optional part. So many times you will have another group attached to the phosphate like we have right here. Usually these do have some nitrogen in them, or they do have maybe a charge or a lot of polar covalent bonds. So that's the general phospholipid structure. If we look at the details of what we end up with, we do have these long fatty acid tails like we're seeing right here. And the important thing to remember about them is that they are very nonpolar. So when we say nonpolar, this is referring to the fact that they are made up of nonpolar covalent bonds. And since they're made up of nonpolar covalent bonds, we could also say that they are going to be very hydrophobic. So that is a property that we want to remember about the tails of the phospholipids. Now on the other end of the molecule, where we have the glycerol backbone, the phosphate, and then this other optional group, we are going to have lots of polar covalent bonds. So this area here, we want to remember is going to be polar. And because it is polar, it is going to be hydrophilic. So when all is said and done, we have two ends of the molecule that are really behaving very differently from each other. And we could call that amphipathic, or amphiphilic would be another term that would describe that. And again, that's just showing that we have two ends that are behaving in opposite ways. When we draw a simplified version of the phospholipid, we tend to draw just a little circle for what we call the hydrophilic head. And then we have two lines that come down, and that would represent those fatty acid tails. Okay, so up at the top would be the head group, which is the hydrophilic part. So if we go back to this first picture that we looked at, where we see a picture of a membrane, notice that there are a lot of phospholipids here in this membrane. And these again are shown in blue. When they form the membrane structure, we're going to end up with what we call a bilayer. And so this is a phospholipid bilayer, meaning that we do have two layers of phospholipids. And when we get those two layers, what's always going to happen is that the inside of the membrane is going to get all those fatty acid tails. And because we have all the fatty acid tails inside the membrane, that area, or the core or inside of the membrane, is going to be hydrophobic. Again, you could also say that it's going to be nonpolar, because hydrophobic things tend to be nonpolar, or we also know that those fatty acid tails are made up of a lot of nonpolar covalent bonds. And then on the other two ends, so on the top and then also on the bottom here, that's where we're going to have those head groups, and those are the parts that are going to be very hydrophilic. This is important because inside the cell, which in this picture is going to be down on the bottom, so we can think of all of this down here as the cytoplasm side. Cytoplasm has a lot of water in it, so it is an aqueous solution, and notice that the hydrophilic part of the membrane, what would like water, is going to be exposed to that cytoplasm. And then on the top, which is going to represent the exterior of the cell, there's very likely going to be a lot of water surrounding that as well. So we do have those hydrophilic head groups that are going to be exposed to the water on the outside of the cell. So every time we have a membrane formed, regardless of where that membrane is going to be, we are going to have two layers of phospholipids, and the phospholipids will always be oriented so that the hydrophobic tails are on the inside, and then those hydrophilic head groups will be the outside of the membrane. While the phospholipids are going to form the main structural part of the membrane, the proteins are more important for the function of the membrane. And so these are what's going to give the membrane its unique characteristics. This is what will make the plasma membrane different from perhaps the membrane that you have around the nucleus or around the mitochondria, endoplasmic reticulum, all those other membranes that you have inside the cell. So recall that proteins are made up of amino acids, and those proteins can take on many different shapes and they can perform many different functions. And that is the case for the proteins that we have in the membrane. So in our diagram, we're going to represent them as green structures. Here we have two examples of proteins associated with the membrane. And these proteins are going to fall into two major categories. This one here would be what we call an integral protein. And we're calling this an integral protein because it does penetrate or run through the hydrophobic core of the membrane to some extent. Now, in this case, this happens to be one that spans from one side of the membrane all the way to the other side. This would be what we would call a transmembrane protein. And a transmembrane protein is really just one type of integral protein. So we can have integral proteins that just penetrate that hydrophobic core just a little bit. The other protein example that we have on here, this one is going to be what we would call a peripheral protein. And this is peripheral because it does not touch that hydrophobic core at all. So this one is just loosely bound to the surface of the membrane, and it is just going to be touching those hydrophilic head groups. Now, anytime we have something associated with the membrane, such as these proteins, it is very important to understand that the proteins have to really match the area of the membrane that they're touching. So they have to have similar characteristics. So if we looked at the integral protein example that we have on this slide as an example, notice that a portion of it up here and a portion of it down here is right next to those hydrophilic head groups. So because it's next to them, the only way that it could be next to them is if that section is also going to be hydrophilic. So that portion or those portions of that particular protein, they will be hydrophilic. And then on the inside, this section here, notice that this is actually touching the hydrophobic tails. So again, it would have to match that, otherwise it's not going to insert in the membrane as we see it here. So the transmembrane proteins will definitely have sections that are hydrophilic and then other sections that are hydrophobic. And the peripheral protein, because it is just loosely bound to the outside of the membrane, it is going to be touching those hydrophilic head groups, and so it is going to be hydrophilic on the outside. Proteins can be really large structures, so when we're talking about it matching the characteristics of the membrane areas that it's touching, we're really just talking about the surface of the protein. So what's buried on the inside, so the section that's in here, if this was considered kind of a barrel-shaped protein, the inside of that barrel may have other properties to it, but the outer surface of it is going to match the membrane structure. The third molecule that we haven't talked about yet, these are the carbohydrates, and the carbohydrates are shown here in the pink or in the purple that you see. And the carbohydrates are going to be very important for distinguishing cell types from each other. That's one of their major characteristics. These carbohydrates will be found on the exterior side of the cell, So this, as it is drawn, is specifically a plasma membrane. And these carbohydrates, they do come usually in two different forms. We have some that are attached to lipids, and that is this first example here, because we do have it attached to some fatty acid tails on the other side. If they are attached to lipids, we call them Glyco Lipids. And glyco does refer to sugar, so this is basically a sugar lipid, which it's a lipid that has sugar attached to it. The other type of carbohydrate would be the one that we see here, and this one is attached to protein, so this is going to be what we call a glycoprotein. So again, you will have carbohydrates many times associated with the exterior side of the membrane. They are going to be attached to lipids or proteins, and these are going to be important for recognition between cells and to help cells distinguish one another.